Hi, I'm Jan Witkowski from Cold Spring Harbor Laboratory, and this is the occasion of the 89th Cold Spring Harbor Symposium on Quantitative Biology. And the topic this year is senescence and aging, and I'm delighted to have Margaret Goodell with me. And I've completely forgotten where you're from, where you are at the moment, Margaret.
Well, I'm at Baylor College of Medicine in Houston,
yes.
Texas.
Okay.
Mm-hmm.
So I haven't yet had the benefit of hearing your talk. So perhaps you could begin by just giving us a sort of a brief outline of what you're going to be saying.
Sure. I'm going to talk about clonal hematopoiesis. And this is a phenomenon that was discovered really only about 10 years ago and has really revolutionized the way many of us think about blood function as we age. And in fact, the same phenomenon happens in other tissues as well. It's the concept that your blood cells are always, the stem cells that make the blood really, are always acquiring mutations. Mm-hmm. And over time, that creates a lot of diversity in your blood. But over a longer period of time, some of those mutations have beneficial effects on the stem cell. And those stem cells then outcompete the other ones over the 60, 70 years of a normal lifespan, or 80 years of a normal lifespan. And so your blood and bone marrow doesn't quite become clonal, that there's one cell, but there's a much smaller number of cells that are producing all the blood. So that state of clonal hematopoiesis is known to have a higher level of risk for hematologic malignancies, but it also appears to affect many other tissues and increase all-cause mortality. In other words, if you have this state at a certain age, you're more likely to die of a variety of other causes. Mm-hmm. And the work I'm going to talk about today is how we think it's interacting with the cardiac muscle to promote cardiac disease with age.
So in what way are these mutations beneficial? Because one always thinks of mutations as being deleterious, but of course I suppose you're beginning with a large population of cells.
Right,
so,
exactly.
So just by numbers, some mutations may be beneficial.
Exactly, and this is what is fascinating to me as a stem cell biologist about this whole phenomenon, because you have about 200,000 stem cells when you're young, and all of those are acquiring mutations at a rate of about 10 mutations per year. And so each individual stem cell may not have that many mutations over time, but if you look at the whole pool, all of your genes are mutated multiple times, even by the time you're 50 years old. And so you can have mutations in genes that are deleterious and that stem cell then disappears, or most mutations are neutral, and about 20 or so have a beneficial effect. They may extend the lifespan, it may make it more resilient, and so in the face of various insults, it could be inflammation, it could be toxic agents that we ingest or are exposed to, and that small subset of stem cells then ends up having an advantage over time and sort of wins this race.
So how many of those clones are there? Do you have any idea?
Well, I mean, it kind of depends on when you look. So if you look in a 20 or 30-year-old, I mean, in a way, by even that age, because they've already accumulated many mutations, hundreds of mutations in each individual stem cell, there are many, many clones still. They have 200,000 different clones. But by the time people are in their 6th and 7th decade of life, it seems to have winnowed down. A lot of stem cells are lost through various attrition, and we can say, I mean, people have anywhere from 10 to maybe 100 really active stem cells when they're older.
And you talked about this sort of all-cause mortality. So is there any indication of how that comes about?
Well, that's something the field is very interested in, and that's something that I'm going to talk about. And we think that at least some of these mutant or variant stem cells not just grow better, but some of them are, let's say, hyperinflammatory. And so then they make progeny, let's call it macrophages, T cells, whatever. They're making all these progeny that are then going into the blood, and then they are infiltrating all of these other tissues. So if you have a more inflammatory downstream cell, a macrophage, then it's like interacting in the liver and the pancreas or whatever. It can create damage in those tissues.
It's interesting. I wrote a couple of papers on early ideas about cancer, and inflammation as a cause of cancer was very popular at the turn of the 19th century. Anyway, so coming back.
It's coming back.
All the good ideas always
come back. Of course, yeah.
So turning on to your specific work on cardiac muscle, what are you doing?
Well, we have a great collaboration with somebody at Baylor, Na Li, who is an expert on atrial fibrillation. And so we've been making a mouse model with this clonal hematopoiesis, a specific flavor of the clonal hematopoiesis. And then we leave those mice on the shelf for a little while and let those cells circulate in the blood, and then we give them to her, and she does all sorts of cardiac function tests. And we can see that this, in the context of this clonal hematopoiesis, it seems to have a negative effect and reduces the contractility of the heart and also increases atrial fibrillation.
And do you find inflammation in these hearts?
We do. We think this particular flavor of clonal hematopoiesis is sort of very inflammatory, and we think that contributes to the dysfunction in the heart. And when we do various tricks to knock down the inflammation, we can at least partially correct the phenotype.
And this is a direct effect on muscle cells, on the does it have any effect on satellite cells?
Yeah, so it is a direct effect in the sense that we can manipulate a small number of things and see a big effect on the cardiac muscle. However, if you think about it, it's incredibly complex. Even in the heart, it's mostly cardiomyocytes, but there are endothelial cells, there are blood cells, there's a whole bunch of other interstitial cells there, and fibroblasts as well. And in the blood, you have this whole panoply of cells that are circulating. And so it is a little bit hard to say exactly which cell is doing what to what cell, because all these things are circulating, and even the circulating cells are also releasing cytokines and other factors that then in turn can affect these other tissues. Right. So I think in general, that's something that the field is really interested in trying to solve, and it may be different for different tissues, right? There may be effects on the lung and the pancreas and so on, and a number of labs have shown effects on different tissues, and the mechanisms might be slightly different in different contexts.
Actually, I realize I've just been confusing cardiac and skeletal muscle when I talked about satellite cells. Yeah. Coming back to the hematopoietic system, can you isolate these deleterious? No, these they are deleterious.
The variant
that's better.
Yes.
Right, but can you isolate them? And
it's difficult to isolate them because the remarkable thing about them is that they have this mutation, but they end up kind of becoming better stem cells, and most of them look exactly like a normal stem cell, but they behave a little bit differently. So there's no good marker yet that anybody has found, right? But people are approaching it by doing various single-cell sequencing, where you can get at the genome mutations at the same time, and then you can really look to see what the mutant cells are doing and what the non-mutant cells are doing in the same blood sample by various single-cell sequencing tricks, basically.
Let me ask, why did you choose the heart as a organ to study as opposed to liver or lung or kidney or some other?
I think in all of science, it's important to have great collaborators, and you look for somebody who you can work with, who is the right match culturally, and who is willing and has somebody else in their lab who can work on the project as well. And in my collaborator Na Li, we found that, and it's been a really, really nice collaboration between my graduate student and her postdoc, who've met basically very, very frequently in order to conduct all these experiments.
So it was a sort of a pragmatic choice rather than the features of the aging heart that
it is both. I mentioned that clonal hematopoiesis is associated with increased all-cause mortality, right? And probably the biggest player is cardiovascular disease, and so it is a big target. It is something that has potential impact clinically, but it's also the convenience and having a good match with a good collaborator.
Sure.
Yeah.
So the sort of $64 question, of course, is always, is this, well, I'm sure you're hoping, but to develop therapies that might alleviate these problems?
Yes. Again, a number of people in the hematology field are really fascinated by clonal hematopoiesis in general, and it seems that, let's call it roughly 20 different mutations can lead to this this behavior, and there's somatic mutations in the somatic cells, right, that are acquired over time. And a lot of people are thinking about how to interfere with those, mutant cells and bias the blood production to maybe a less deleterious cell. Mm-hmm. I will say it's tricky because the cells aren't like terrible. It's not a cancerous state, right? And you're not going to die of clonal hematopoiesis, but it can lead to all of these other conditions. And so the field is really sort of having some existential moments about, well, if you're not going to die of it, how aggressively do we treat it? And are there things that won't cause secondary effects that you could use? And to what extent just living a better lifestyle, a healthier lifestyle, will that kind of ameliorate it?
Yeah, that's a social problem, which I think is much more difficult to cope with, deal with than the sides that's involved. Well, I'm afraid we're going to have to stop because people are going to be flooding back into the auditorium. But anyway, Margaret, thank you very much.
Thank you very much for having me. It's always a pleasure to be here, and I've enjoyed it. Yes.
Thank you.
You.